this week. Okay, so something I wanted to mention is that my notes for this unit I couldn't find in my drive. I don't think they exist. I must not have typed them. So I do have them written down. I'll ask you guys, do you think you want me to post these so you can reference them, or do you think it'll be helpful? Okay, so I'll post these instead of the typed notes. So that's why there are no typed notes this time. And I also have a list of just like terms from this unit. So I'll post that too, but it's not a comprehensive like study guide. Okay, so for the first section, I'm just gonna do kind of like a broad overview of the digestive system. And then in the second part, I go into like insulin and like uptake of glucose into muscle, liver, and beta cells. Okay, so the two main GI functions are motility and secretion and absorption. So then there's macro scale and micro scale motility. Macro scale peristalsis is just like propelling the food throughout the system, because it's kind of like one tube, so it helps push the food down. And then segmentation is mixing of the food throughout, moving it through the system. So this says proximal contraction and distal relaxation for peristalsis. So that just basically means that the tube is contracting here and relaxing here, which creates a pressure gradient pushing the food forward. Segmentation, simultaneous distal and proximal contraction basically means that they're contracting and relaxing at the same time, which mixes the components of the food in between. And then most absorption is done in the small intestine, which we talk about later. So here's another visual of segmentation and peristalsis. And then here's histology. You got 4 layers throughout the digestive system lining. So you have mucosa, and this is where absorption and secretion take place. So you have 3 sublayers, the epithelia, lamina propria, and muscularis within the mucosa. And then there's submucosa, which kind of just holds the layers together, and it's made of connective tissue, nervous and lymphatic tissue. And then muscularis is smooth muscle and nervous tissue, and you have the interna and externa. Something to note is the myenteric plexus. So this is a layer of nervous tissue. So the nervous system innervates the digestive tract. And then there's serosa on the outside, which is just a connective tissue wrapper. And here's a visual. In this case, we're looking at the stomach, because the layers will vary based off of where you are in the digestive system. So you can see like the mucosa with the epithelia on the outside, and you get into the muscularis, submucosa, and serosa. And then towards the right, there's a gastric pit or parietal cell, which is in the stomach, and that's involved with a lot of secretions that we talk about soon. And like I just said, the composition of these layers varies based off of where you are in the GI tract. So you can see the small intestine has a lot of like villi, which helps increase surface area for absorption. The stomach has the gastric pits and parietal cells. So specialization based off of function. And then here's a general overview of the path of food through the system. Mouth to esophagus, to stomach, to small intestine, to large intestine. And I'm going to go through each of these key organs on a separate slide. So in the mouth, you have the salivary glands, which are exocrine glands. And there are 3, parotid, sublingual, and submandibular. And then it's important to note the different sphincters that separate the different components of the digestive system. So from the mouth to the esophagus, you have the upper esophageal sphincter. And this sphincter is made up of skeletal muscle, which means that it's under voluntary control. A common trend is if something's of skeletal muscle, then it's voluntary control. Like you can consciously control what it does. Smooth muscle is more autonomic, happens automatically. You can't really control if it's open or closed. So this one is like when you swallow, you can control if it's open or closed. So it's skeletal muscle. Then the food travels into the esophagus, and then there's peristalsis, which helps bring the food down into the stomach. And then at the bottom, at the junction between the esophagus and the stomach, you now have the lower esophageal sphincter. And this one is smooth muscle. So you can't control consciously if you're going to open or close that. It's based off of a pressure gradient. So if you have food in the esophagus, the pressure in the esophagus is going to be greater than the pressure in the stomach, which is going to cause the sphincter to open and allow the food to go into the stomach. Yeah.
Basically, so anything involuntary is like something you like can't control. So like that'd be like a heartbeat.
Yeah.
Okay.
Heartbeat is involuntary, but
yeah.
Heart is not smooth muscle; it's cardiac muscle.
Yeah.
But yeah, your heartbeat is automatically controlled. Yeah.
What about like breathing? So like what would that be?
I see what you're getting at, but
yeah,
like
I know you can control breathing, but it's like
those are more like breathing and heart rate are regulated by like the brainstem because they're under like nervous control. But when it's directly, when you have a muscle controlling a function, if it's skeletal muscle, it's something that you can consciously control. And if it's smooth, you can't consciously control it. But the nervous system also plays a role. Yeah. Yeah, because breathing is like a lot of muscles.
Yeah.
It's not like one. And then the heart is a specific cardiac muscle. So that was a good question, though. And then after the esophagus, we get led into the stomach. And then there are two kind of like ways to look at or divide the stomach into different sections based off of what you're thinking about. So if you're looking at it anatomically or just kind of like the different pieces of it, you have the cardiac stomach, and then the fundus, body, and pylorus or antrum. And then functional divisions, this is just kind of saying they're divided into different sections based off of different like functions. So if you're thinking about it that way, you have the proximal stomach and the distal stomach. The proximal stomach has rugae for storage, and the distal stomach has the oblique layer that has an extra thick layer of muscle that helps in mechanically digesting and like churning the food. Also in the stomach, we have the gastric pits, and this is where the secretions are released. Parietal cells release HCl, and then you also have G, D, and ECL cells that all help regulate the secretion of HCl. So G cells produce gastrin, D cells produce somatostatin, and ECL cells produce histamine. And then you also have chief cells in the gastric pits that release pepsinogen that eventually is converted to pepsin and helps digest proteins. And then at the bottom of the stomach, leading into the small intestine, you now have the pyloric sphincter. And this one is under nervous system control.
And all of this is like in addition to those 4 layers from the beginning?
Yes.
So would these all be in the, like those specific cells, would they be in the mucosa layer?
Yes,
Okay.
because the mucosa is where secretions and absorption happens. So any secretions, so these cells would be considered secreting cells. And then now we're in the small intestine, and there are 3 segments. The duodenum is first, then the jejunum, and then the ileum. And here we have primarily segmentation to get us through that windy tube, but there's also some peristalsis. And then motilin is released during fasting to stimulate a strong peristaltic wave, which is known as the migrating motor complex. And there's villi and lacteals here. And at the end, there's the ileocecal valve that leads to the large intestine. And lastly, the large intestine, the main function here is to absorb water and vitamins. There are 3 main regions, the cecum, colon, and rectum. And within the colon, there's the ascending, transverse, descending, and sigmoid regions. And then there are 2 anal sphincters. You have the internal anal sphincter, which is involuntary, like we talked about before, and then the external one, which is voluntary.
Does segmentation and peristalsis happen everywhere, or just in like the large intestine or the small intestine, large intestine?
That's a good question. I think that it happens everywhere.
Okay.
But in certain regions, like it happens more. Like I said, in the small intestine, primarily segmentation, which is the mixing
Okay.
one, but there's also some peristalsis. Yeah, and like here, there definitely has to be peristalsis to help you like go up the ascending and move through. Yeah.
Probably not as much segmentation because there's not being secreted.
Yeah.
Okay.
So you don't really gotta mix it anymore. Yeah. Okay, now part 2. This one is a little bit more interactive. So we're going to talk about absorption in the small intestine and blood glucose regulation. So to zoom out, to kind of set the stage of what we're talking about, when you eat food, the food gets broken down and is absorbed into the blood, which causes blood glucose to increase. And when the glucose levels in your blood are increased, your body has to do something with it. Like it can't just stay there. So some sort of change has to happen in your body to allow the the glucose in your blood to be absorbed into cells to be used for energy. So insulin is the hormone that's released when there's a high concentration of glucose in your blood, which allows cells to uptake that glucose into them and then use it to make energy. Okay, so I'm gonna pull this up on here. Okay, so in the small intestine, you have, if this works, I have other ones. So this is the lumen. Ooh, sorry. And then, that one's better. This is like the outside of the tube of the digestive tract. So then over here is going to be the blood. And then this is the lumen. So this is where your food is after you eat, and it's in the small intestine. So we have 2 transporters on this. This is the apical membrane, and this is the basolateral membrane. On the apical membrane, we'll put two transporters. So this one down here will be GLUT5. Did you guys talk about this yet?
I remember seeing it in the video.
Did you do it in class?
Not yet, but it was in the videos.
Okay. That's perfect. Because then you probably won't go over it. And then SG, that's SGLT1. So do you guys know what the simple sugars are, the three simple sugars? What are they?
Glucose, galactose,
Mm-hmm.
and fructose?
Yeah. So after we've secreted all the things in the stomach and like started digesting the food, the idea is that the food will be in the form of those three simple sugars now that we're in the small intestine, so they can be absorbed into the blood. So
The enzymes that would break them down before they get there in the small intestine, would that be in the stomach?
yes. So now two of those simple sugars, glucose. This marker is terrible. I'm gonna use blue. So that's glucose. And galactose are part of your food. And then you have fructose. So these all need to get absorbed into the blood. And they do it through these different transporters. So we're going to talk about GLUT5 first. It's selective for fructose. So fructose can go right through GLUT5, I'm just going to call it that, by facilitated diffusion. So that means that it's going from a high concentration gradient to a low concentration gradient through the transporter. Glucose and galactose both go through SGLT1. Into the cell lining the small intestine through secondary active transport. Basically what that means is by allowing sodium to move into the cell down its concentration gradient, glucose and galactose can use the energy that it took to move sodium down its concentration gradient to move into the cell.
So sodium needs to be there in order for glucose and galactose to move through.
Yes.
And let's say if like fructose were to try to move through the SGLT, like it would just like get stuck or just like it just wouldn't like answer at all.
It wouldn't be able to move through because there needs to be energy to get these cells to get it through, and the sodium is what's generating the energy because it's being allowed to move down its concentration gradient, which powers the ability for these two sugars to be able to come into the cell.
So fructose is like the only one that just doesn't need anything to move through.
Yeah, it's facilitated, so it's moving just.
Wouldn't those other sugars be able to move passively if the lumen is where they're, like if fructose is able to, are they able to?
That's a good, as I'm like explaining this, I'm like, that would make sense. But I have that as secondary active transport. So I'm gonna look it up once we finish the drawings because the concentration should be higher in the lumen. So shouldn't they move down passively? Yeah, I agree. So I'm gonna look it up. But for now, it's secondary active transport, so that's what I have in my notes, so I'm assuming that that's what it is. Okay, so now we're in the small intestine cell, and we want to still get into the blood. So there's another transporter over here that I want to use blue for. Oh, also, the name of it, it's like sodium glucose something transporter. So they will help you remember.
Okay.
Sodium glucose galactose transporter, maybe? I don't know. Yeah, but the name will help. I'll pull up the name too. And then again, all three sugars can go through GLUT2 by facilitated diffusion. Because now that they're in the cell, the concentration is going to be higher than in the blood. So all three sugars move through GLUT2 via facilitated diffusion into the blood. So now we have glucose, galactose, and fructose in the blood, circulating in your bloodstream. So here's the drawing that I have on the board up on the slide. Okay, so here's what the names of them mean. SGLT1 is sodium glucose cotransporter. And it uses secondary active transport. Yeah, sodium. Okay. Does that make sense? Okay. And we'll keep that there and we'll look that up. So here's it written out in words, everything we just talked about. So now glucose is in the blood, and now blood glucose levels are increased because we just ate food and the sugars are in our blood. So now the glucose is going to travel through the blood and reach the pancreas, which have beta cells. So beta cells are just special cells in the pancreas. And the beta cells are going to release insulin, which is a hormone that's released in response to high blood glucose. So that makes sense. We have the high glucose, blood glucose. We go to the pancreas, which is then going to release insulin. And what the insulin is doing is kind of signaling to your body that there's glucose in the blood and we need to take it into the cells. Because we can't just keep raising our blood glucose because we're going to move out of homeostasis and we want to make energy. Okay, so now we have the next drawing. Okay, so paint. Now we're at, I probably should start over here. So this is just a beta cell in the pancreas. So it's just a cell in the pancreas. Slowly we'll. Beta cell in the pancreas. And again, like we had on the basolateral side of the small intestine, we have GLUT2 on the beta cell. So when glucose arrives at the pancreas through the blood, do you guys have any guesses what it's going to do? Yeah, because like we saw over here, GLUT2 allows facilitated diffusion of glucose. So now in the pancreas, we have a high concentration of glucose in the blood. So glucose is going to go through GLUT2 via facilitated diffusion. So now we have glucose in the beta cell. And it's going to be transformed into ATP by cellular respiration. But you guys don't have to know that. Basically, you just have to know that glucose gets converted into ATP in the cell. If you have to take biochem, that's like what you do in biochem, is learn how that happens. And then what ATP does here, it's a different color, is it serves as a ligand to block this potassium channel. So then ATP comes over here. This serves as some sort of signal to block this. And what that does is it basically creates a depolarization. Or it changes the voltage in the membrane, it changes the concentration of ions in the cell, which causes this channel over here, which is a voltage-gated calcium channel, to open. Allowing calcium into the cell. And then if you remember from, I think it was last semester, calcium likes to dock to vesicles and help them exocytosize, if that's what you say, like we learned about in neurons. So calcium comes over here, docks to this vesicle, and brings it to the membrane so it can let the contents out into the environment. Does like the sequence of events make sense here? Okay, so do either of you have any guesses what this thing is that it released?
Insulin.
Yeah, exactly. So it's kind of cool how it works. Like, the glucose arrived at the pancreas, all this stuff happens in the cell that lets it release insulin into the blood. So now we have insulin circulating in the body. And then the next two drawings are going to talk about what insulin does when it arrives at tissues. So I have the same thing right here with some captions so you can remember what we talked about. So now that insulin has been released by the pancreas, metabolic cells, are basically cells that could use the glucose, respond to increase their uptake of glucose, to use it to make ATP. So metabolic just means that they're capable of doing cellular respiration to make ATP and use the glucose for energy. And glucose uptake happens differently if you're in skeletal muscle or liver cells, which we'll talk about here. So do Do you guys have a preference as to which one I take away? No? Okay. I'll take away this one, but I do want to look up how the secondary active transport works with that, if you guys can remind me. Okay, so now the insulin travels to skeletal muscle, or we can go two places. So first, we're going to talk about skeletal muscle. So the insulin goes through the blood from here. Now we're at skeletal muscle. And on the skeletal muscle, there's a transporter. We're going to do it in green. Called, it's a different one, GLUT4. And this one basically does the same thing as GLUT2. Allows glucose into the skeletal muscle cell. Oh wait, no it doesn't. Completely disregard that, hold on. Okay, no. We do have something different going on here. So GLUT4 is different because it's not located on the membrane. GLUT4 is inside the skeletal muscle cell, just chilling. It's just hanging out. But on the skeletal muscle membrane, there is a receptor. And that receptor receives the insulin signal from the blood, which then triggers GLUT4 to dock to the membrane. So insulin binds to the insulin receptor, some sort of cascade of events happens that triggers GLUT4 to dock to the membrane, and now it's here. So that's the difference between GLUT4 and GLUT2. GLUT4 now can allow glucose into the cell via diffusion. And now glucose is in the skeletal muscle cell and can be used to make ATP and power contraction. So that one's not too many things going on there. Does that make sense? Okay. Do you have a question? Oh, okay. So here's that one. Oh, and I forgot to make it so it appears. But now we'll go into the liver, which is not too, the worst one is like understanding the beta cell, I'd say. And then 2 in the liver. So that's just like a cell in the liver. Again, we have GLUT2. On the membrane, so glucose, once it arrives, can just diffuse right in to the liver cell. But here we have a hexokinase enzyme. So hexokinase is going to phosphorylate glucose. So then it can be incorporated into glycogen by glycogen synthase. So I'm just going to write hexokinase. And that's an enzyme. And then plus glucose goes to, which is just glucose phosphate. And then with glycogen synthase, it can be converted into glycogen, which is just the storage form of glucose. So I feel like just visualizing it helps instead of just like trying to memorize like GLUT2. Now you can see like GLUT4 docks to the membrane. GLUT2 always allows facilitated diffusion of glucose into the cell. Or in the case of the small intestine, into the blood. So here's that one. I was going to change this drawing. GLUT2, I wanted to make it blue like it is on all of the other drawings. So it's the same GLUT2, and I'm going to fix that. I made it orange. Okay, so I have a few questions that correspond to this. Oh, it's a login. If I can get in. Oh. I can't attempt to log in for 30 minutes. Wait, why did that pop up like after I did it like one time? Wait, maybe. Let's see if I can do it on my computer or something quickly. Because then I could open the questions. Okay, I'm in on my computer. So I bet I could plug this. Okay, the first one is up. And it is GLUT2 transporters transport glucose via I'm looking up.
I feel like there's a sodium potassium pump on that other membrane,
Mm-hmm.
the basal membrane, that is taking sodium out to bring potassium in. So maybe that's why, like, sodium is moving down its concentration gradient, giving like power to that pump.
Yes, but why would glucose and galactose need energy,
Yeah.
right?
I have no idea.
If you're right, though, about the sodium. My thought is that the cells in the small intestine need glucose to function too, so they keep some glucose there, maybe. But I'm gonna keep looking. So we'll go over this question. So we have facilitated diffusion and secondary active transport. So one of the transporters does do secondary active transport, but it's not GLUT2. Do either of you remember which one?
That's the SGLT5 one.
Yeah. So that was the SGLT one that's in the small intestine that allows the glucose and galactose to go into the cell with the input of sodium down its concentration gradient, which we're still trying to figure out.
Wait, so GLUT2 is, would that be secondary active transport?
GLUT2 is facilitated diffusion.
Okay, so like, I'm just having trouble like figuring out like the difference between like active transport and secondary active transport.
Yeah. So none of these use active transport, so you don't have to worry about that. But if you remember, active transport uses energy, which is ATP, to pump something against its concentration gradient. Secondary active transport is active transport because it still uses energy, but instead of directly using ATP, it uses ATP formed by something else, basically, to power the process. So in the case of SGLT, I'll just draw it small here. So we're in the small intestine. SGLT is right here. Glucose and galactose are out here. And apparently are at a higher concentration also in here. So in order to get these in here, you would need to use active transport, but we're not going to directly use ATP. Instead, sodium is at a lower concentration inside the cell. So we're going to allow sodium to move down its concentration gradient into the cell, which then powers the process of allowing these two to come into the cell. So instead of using energy, we're using kind of like this. This is, having this concentration gradient is kind of like a potential energy in a form. So allowing this to go down then powers the movement of these two sugars into the cell, is what secondary active transport means.
Oh, I see that.
So that's SGLT1. All of these ones, the GLUT2 here, over there, and then we also have a GLUT2 here. Use facilitated diffusion, which means that they're not using energy, and they're just letting whatever it is diffuse down its concentration gradient through a transporter.
I found in the top hat reading that the SGLUT transporter is important after a low glucose meal when the concentration of glucose is not sufficient for effective transport using the GLUT transporters.
Using the co-transporters?
Yeah, the regular like GLUT 2 and 5.
Okay.
So that's why it needs the energy.
Okay, so it's after a low meal, so there's not a high enough concentration gradient difference, so it needs to use energy to push it into the cell.
Yeah.
Okay. So based off of that, we also have, if you remember, this one's GLUT5. And this one we said moves fructose down its concentration gradient. So what I have in my notes is that SGLT works for glucose and galactose, while GLUT5 works for fructose. Based off of what you're saying, there might be another transporter that moves glucose and galactose in.
It looks like there's GLUT1, 2, and 5, which are all glucose facilitated transporters. And then SGLT is an active one. It looks like there's a whole bunch. I bet it just doesn't go into that much detail for lecture.
Right.
Like these reading assignments are like extra credit almost.
Yeah.
So they probably just do that to like help simplify it for people.
That's exactly what I was going to say. I think for the purpose of this course, you probably just need to remember SGLT, secondary active transport for glucose and galactose. But technically, since we're like talking more deeply about it, we're like, why does... So, but I think this is all you need to know.
Yeah.
But that makes sense, so good to know. We figured it out.
Mm-hmm.
Okay, the next one is up. GLUT2 transporters are located, and then you can select any. Bless you. Or did you cough? Oh, sorry. Okay, so just so we have time to get through all of them, we agree that GLUT2 is on the beta cells, but GLUT2 is also here in the liver that allows glucose in passively, and in the small intestine, allowing all three sugars into the blood. What are the three simple sugars that are absorbed into the bloodstream through GLUT2? Nice. Okay. Glucose, galactose, and fructose are three simple sugars. So you guys have to know the other three that are like the disaccharides.
I'm not sure. I know he mentioned them through the videos.
Okay. I think I would know this going into the next exam.
Okay. Maltose, sucrose.
Yeah, here they are. Oh, it's not up there. But it is sucrose, lactose, and maltose are the disaccharides, so that's just two simple sugars together. Okay, the next one's open if you want to try it.
So by the time sugars get to the small intestine, they're monosaccharides. Does that mean by the time they get to the stomach, they're disaccharides?
No, I wouldn't say that, no.
Okay.
Because that would mean that your food is already into disaccharides by the time it's gone down the esophagus.
If the salivary amylase is like working at that though.
I would say maybe some of it is, but it's not perfect. It's not like all the sugars here in the small intestine are simple, and all of them are disaccharides once they get to the stomach. Because also the salivary amylase, like different enzymes, digest different types of food.
Okay.
So I think only protein is digested in the stomach, if I remember correctly, because of the pepsinogen and the pepsin. So SGLT is this one here that we talked about with glucose and galactose going up their concentration gradient using the diffusion of sodium into the small intestine cell. So that one is going to be secondary active transport. Does that make sense, or are you still confused a little?
Yeah, I started like just like worrying because like I just watched the videos like say like this morning. So like,
Okay.
yeah, so,
Still got to sit a little. Maybe, yeah. Do you know what do you understand facilitated diffusion, the concept of that one? Or do you want me to go over that really quick?
Yeah, probably.
Okay. So diffusion means that it's going from a high concentration to a low concentration. So it's allowed to do it without the help of anything. So you know osmosis?
Yeah.
Water moving, so that's a type of diffusion. If it's active, it's going against this concentration gradient. It needs some sort of help. So in this case with the glucose, in the fed state, I'm going to do it like this. We have so much glucose out here in the blood. So it's high. There's a high concentration of glucose in the blood. The concentration of glucose in the liver is low. Like there's nothing in there. We haven't eaten in a while or something. So glucose wants to go into the liver cell. It wants to diffuse into the liver cell, but it can't physically go through the membrane of the liver cell because it just doesn't work. It can't. It needs a transporter. So it's diffusion, but it's facilitated because it needs the help of the transporter. It's moving down its concentration gradient, but it has the GLUT2 transporter. Here, it's the opposite. We have a high concentration of glucose in the intestinal cell. We need to move glucose. There's a small concentration here. We need to force glucose into the cell, and it can't do that by itself. So allowing sodium to go into the cell powers this transport of glucose up its concentration gradient, which is why it's active, or secondary active transport in this case. So hopefully, as that sits, it might make more sense. Okay, next one. What hormone is released in the fed state? What hormone is released in the fasted state? So today we talked a lot about the fed state. We didn't talk about the fasted state. There's a whole nother process with a different hormone that's released to try to do the opposite, increase blood glucose when it's decreased because you haven't eaten food to maintain homeostasis. So that first one is correct. Insulin is released in the fed state, like we talked about from the beta cells, and glucagon is the fasted hormone. The purpose of segmentation is to I think I might have messed this one up. Yeah, I did. Okay, so the purpose of segmentation is to mix food. You guys got it? Where are the gastric pits located in the GI tract? Perfect, yeah, the gastric pits are in the stomach, and they have a lot of cells that are involved with secreting enzymes to break down food. Last one. What are the accessory organs in the digestive system? Select all. So we didn't talk about the secondary organs as much today. But they're also important to review. Nice. So the pancreas and gallbladder are definitely two of the accessory organs. The liver is the third one. The colon is a region of the large intestine, which is just a part of the GI tract. It's not an accessory organ. Okay, so I'm going to go back to... Okay, we're done, but I just wanted to share. Here's my study guide from this unit if you want to use it. Yeah, I recommend like breaking it down because this can be overwhelming into like absorption in the small intestine and then like regulation. I even have on the side a list of enzymes that could be helpful. Yeah, so feel free to ask any questions if you guys have any.